Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with synthesis of triglycerides. In today's session, we will start with metabolism of adipose tissue, in which we have adipose tissue under a well-fed state, fasting state, by diabetes mellitus and obesity. Then we have adipokines, in which there is leptin and adiponectin. Lastly, we have white adipose tissue and brown adipose tissue. Good morning, everyone. Let us continue the topic. Metabolism of adipose tissue. The adipose tissue serves as a storage site for excess calories ingested. The triglycerides stored in the adipose tissue are not inert. They undergo a delayed turnover with new triacylglycerol molecules being synthesized and a definite fraction being broken down. Coming to adipose tissue in well-fed conditions. Under well-fed conditions, active lipogenesis occurs in adipose tissue. The dietary triglycerides transported by chylomicrons and the endogeneously synthesized triglyceride from the liver brought by VLDL are both taken by adipose tissue and are esterified and stored as triacylglycerol. The lipoprotein molecules are broken down by the lipoprotein lipase present in the capillary wall. In well-fed conditions, glucose and insulin are high. GLUT4 in adipose tissue is insulin dependent. The insulin increases the activity of key glycolytic enzymes as well as pyruvate dehydrogenase, acetyl CoA carboxylase, and glycerol phosphate acetyltransferase. A stimulant effect of insulin on the uptake of glucose by adipose tissue, then glycolysis on the utilization of glucose by HMP pathway, and enhances lipogenesis are seen. Insulin also causes a decrease in the hormone sensitive lipase, and so lipolysis is decreased. In this diagram, we can see the adipose tissue in well-fed conditions, where insulin is stimulated by insulin stimulates glucose, VLDL, and chylomicrons from the gut and enters into the fatty adipose tissue. It also stimulates the release of triacylglycerol. Coming to adipose tissue in fasting condition, the metabolic pattern totally changes under the conditions of fasting. Triacylglycerol from the adipose tissue is mobilized under the effect of hormones, glucagon and epinephrine. The cyclic AMP mediated activation cascade increases the intracellular hormone sensitive lipase. The phosphorylated form of enzyme is active, which acts on triacylglycerol and liberates fatty acids. Under conditions of starvation, high glucagon, ACTH, glucocorticoid, and thyroxine have lipolytic effect. The released free fatty acids are taken up by peripheral tissues as fuel. In this diagram, we can see adipose tissue in fasting state. Under the fasting state, growth hormone is secreted by pituitary gland, and cortisol and catecholamines secreted by adrenals enter the adipose tissue and bring down the lipoprotein lipase levels. This is a classification of well-fed condition versus fasting condition. Under a well-fed condition, the lipogenesis is increased, whereas during fasting state, lipogenesis is inhibited. During well-fed state, the lipolysis is inhibited, whereas during fasting, lipolysis is increased. During well-fed state, lipoprotein lipase is active, and Insulin inhibits HS lipase, whereas during fasting, glucagon activates HS lipase and free fatty acids in the blood increases. Coming to adipose tissue and diabetes mellitus, lipolysis is increased and increased free fatty acid level in plasma is noticed in diabetes mellitus. The insulin acts through receptors on the cell surface of adipocytes. The receptors are decreased, leading to a increased resistance in insulin resistance in diabetes mellitus. In type 2 diabetes mellitus, there is increased resistance and the different insulin signaling pathways are affected differently. Hepatic gluconeogenesis occurs uninhibited during, leading to hyperglycemia. However, increased mobilization of free fatty acid from the adipose tissue and the persistently high free fatty acid level in the presence of hyperinsulinemia stimulates the synthesis of triacylglycerol. The overproduction of triacylglycerol to increased release of free VLDL from liver causes hypertriglyceridemia. The excess deposition of triacylglycerol in adipose tissue accounts for the obesity prevalent in type 2 diabetes mellitus. Coming to adipose tissue and obesity, the fat content of the adipose tissue can increase to unlimited amounts depending on the amount of excess calories taken in. This leads to obesity. Plasma insulin level increases, but the insulin receptors are decreased and there is peripheral resistance against insulin action. When the fat droplets are overloaded, the nucleus of adipose tissue cell is degraded. The cell is destroyed and triacylglycerol becomes extracellular. Such triacylglycerol cannot be metabolically reutilized and forms the dead bulk of obese individuals. In this diagram, we can see that in normal persons there is lean adipose tissue, whereas in obese persons the size of the adipocyte increases, leading to deposition of triacylglycerols. Coming to adipokines, that is adipose tissue derived hormones. Adipokines are a group of active factors involved in maintenance of energy homeostasis as well as resistance to insulin. The important adipokines are leptin, adiponectin, resistin, tumor necrosis alpha, and interleukin 6. Coming to leptin. Leptin is a small peptide produced by adipocytes. Leptin receptors are present in specific regions of the brain. The feeding behavior is regulated by leptin. The defect in leptin or its receptor can lead to obesity. Leptin also prepares body to adapt for starvation. Leptin regulates energy balance and exerts an insulin sensitizing effect. Low levels of leptin increase the occurrence of obesity. In this diagram, we can see that leptin, when released out of adipose tissue, enters into the brain. Then from the brain, the food intake increases and there is decrease in the energy expenditure. Coming to adiponectin. Adiponectin is another polypeptide which increases the insulin sensitivity and muscle and liver exerts an anti-atherogenic effect. The decreased levels of adiponectin will increase the atherosclerosis. Also, the decreased levels of adiponectin are observed in patients with a metabolic syndrome. Coming to white adipose tissue, it is mainly concerned with energy storage. It is made up of spherical cells with very few mitochondria. The triglyceride from of the major component of adipose tissue, that is about 80%, with oleic acid being the most abundant fatty acid. Coming to brown adipose tissue. Brown adipose tissue is involved in thermogenesis. Brown adipose tissue cells are polygonal with more abundant cytoplasm. The brown color is due to the presence of numerous mitochondria. It is primarily important in newborn human beings and adult hibernating animals. Coming to thermogenesis. Thermogenesis is a process found in brown adipose tissue. Heat is liberated by uncoupling oxidation from phosphorylation. So the energy is released as heat instead of trapping it in the high energy bonds of ATP. Coming to liver adipose tissue axis. Liver produces fatty acids and triacylglycerol, which is transported by VLDL, that is very low density lipoprotein in the blood. The fatty acids from VLDL are taken up by adipose tissue with the help of lipoprotein lipase and stored as triacylglycerol. The neutral fat is hydrolyzed by hormone sensitive lipase to non esterified fatty acids, which is carried by albumin in the blood. The non esterified fatty acid is utilized by the peripheral tissues, excess of which can be taken up by liver cells. Thus, there is constant flux of fat molecules from the liver to adipose tissue and back. In this diagram, we can see liver adipose tissue axis. Liver synthesizes triacylglycerol. Then, along with help of VLDL, it enters into the muscle tissue and adipose tissue. Then again, in the adipose tissue, it turns back to form free fatty acids to enter into muscle tissue and liver. Coming to fatty liver and lipotropic factors. Fatty liver refers to the adiposition of excess triglycerides in the liver cells. The balance between the factors causing fat deposition in liver versus factors causing removal of fat from the liver determines the outcome. Coming to causes of fatty livers. Causes of increased deposition of fat deposition in liver are mobilization of non-esterified fatty acids from adipose tissue and excess synthesis of fatty acid from the glucose. On the other hand, there is decreased removal of fat from the liver due to toxic injury to the liver, that is secretion of VLDL needs synthesis of ApoB100 and ApoC, and decreased oxidation of fat by hepatic cells. An increase in the factors 1 and 2 or a decrease in the factors 3 and 4 will excessive accumulation lead to fatty liver. Here ends our session. Kindly like, share and subscribe the channel. Thank you.